Hello and welcome to the first of 3 pre-labs we will be doing this semester. So this is on macromolecules. So let's talk about what macromolecules are. So everything living or that has lived previously is made up of what we call organic molecules. Organic molecules are combinations of carbon and hydrogen atoms covalently bonded to each other in many different configurations. We're going to see examples of those shortly. But that's the key. They are also what we call hydrocarbons. And they can also include nitrogen and oxygen and phosphorus and sulfur and other atoms, as we'll see shortly. So macromolecule, if we break that word down into its component pieces, macro means large. So a macromolecule is a large molecule, and they are made up of smaller subunits. And sometimes there are thousands upon thousands of these subunits in one macromolecule. And of these macromolecules, there are 4 major types. And those are carbohydrates, lipids, proteins, and nucleic acids. So let's go through each one of those, and I will describe some of the characteristics of each. So carbohydrates is the first of the 4 types of macromolecules we will talk of. So carbohydrates contain carbon and oxygen and hydrogen, as you can see in the figure on the left, figure 3.1. So we see in this area here that we have carbon and oxygen and all those little hydrogens around it. And in this case, this carbohydrate is glucose, and it is in a ring structure. And it looks a lot like this, and it's got a little antenna that sticks off. And this is an oxygen. So this is our simplest carbohydrate that's really, really important for life. We'll talk more details about that shortly. So carbohydrates are our primary fuel for life. We break down glucose and other carbohydrates to get energy. They are our fuel. And one of the things that's great about them is that they contain a lot of these carbon-hydrogen bonds. These contain chemical energy. This chemical energy originally came from the sun, because plants process the sunlight energy to make that glucose. And we take that glucose and we break it apart and get that energy that originally came from the sun. So they're not just fuel though. There are other types too. There are also structural carbohydrates that build things. And we'll talk about some of those later, the 2 major ones. So we have several different categories as well, based on their size and what they're made of. The first of the categories we're going to discuss are the monosaccharides. Now these are our basic units of sugar. And if we break down this word monosaccharide, we have 2 parts to it. Mono means one, and saccharide means sugar. So these are 1 sugar units. And your book also calls them simple sugars. So if you see the word simple sugars, you will know that this is what they're talking about. Any one of these very simple 1 ring structure sugars. What I mean by 1 ring is that they have carbons and oxygens joined in a single ring, either a pentagon or even a hexagon. They're not always in rings, but these are in this case. glucose, fructose, and galactose are just some examples. There are many other sugars as well. So in all of these, the carbon-hydrogen bonds are broken in order to get that energy. So any one of these bonds is broken and we can get energy from it to fuel our life processes. The most important monosaccharide in humans and animals is glucose. It is used for fueling everything, and anything we eat is mostly broken down into glucose, especially any sort of carbohydrates like starch or even sugars. So most of it we get in our diet from what we just call sugar, which is sucrose. Now sucrose is a bit more complex than glucose, but it's broken down into glucose. We can also find glucose in fruits, but I don't think many of us eat all that much fruit and can get a ton of it from that. But we also get it from things that we eat that are starchy, because starch is made of sugars. We'll talk more about that later. So starchy foods like pasta, bread, rice, corn, etc. So if we have too much blood sugar, which is also called, glucose can be called blood sugar if it's circulating in the blood. If we have too much of it, it can be converted into something called glycogen. Here is glycogen. And it's a temporary storage for sugar. Not long term, just for a little while. And it's particularly cool. So it's a very highly branched molecule. As you see, it branches off in many different ways. And it can continue to branch as it's longer and longer. And it can be thousands upon thousands of glucose molecules long. So let's draw on the right a branched molecule. So let's imagine that each of these circles is a glucose. And here we are in glycogen. Now this is not accurate. It's just a representation of why the branching structure is useful and important. So here's a branched molecule, and this is, we'll call it glycogen. And the way we break it down is we use enzymes. And we'll talk more about enzymes shortly. But enzymes break things down in the body. And I draw them like little Pac-Man because that's useful. Now this little Pac-Man can break down this glucose, but it can only do it one at a time. It can't do a whole bunch, and so it does one at a time there. Now, it can break, there's not just one enzyme, there's lots of copies of the same enzyme that breaks down this glycogen into glucose. I'm trying to stay on screen here. And they can all do it at once, and all break those bonds between it and get that glucose, and then use the glucose for whatever process it needs to be used for. We'll talk about why starch is not a quick burst of energy type thing. So that's what we get from glycogen, is a quick burst of energy. Due to all of those glucoses being broken down at once. Now this is only found in animals, not plants. Plants do not make glycogen. They make something else called starch, which we'll talk about in a bit. So in humans and animals, we store energy long-term as fat. So excess sugar will be eventually converted into fat for storage. So let's talk about complex carbohydrates. And up at the top here it says linking monosaccharides. So complex carbs come in many different forms. So for example, here is that sucrose I was mentioning, what we call sugar, that we put in our coffee and tea. So it is made of a combination of two monosaccharides, glucose and fructose. So when they have a bond together, they're what we call a disaccharide. Di means two saccharides, two sugars. So when we digest sugar, there's an enzyme that will break this bond, and then we can break down the glucose and the fructose into energy. We can break those carbon-hydrogen bonds and the carbon-carbon bonds and produce energy. Now on the right, we see a long chain of what looks to be glucose molecules, and this is what we call starch. So hundreds of glucose molecules bound together in a long single chain. Now it doesn't branch like glycogen. Starch is one long chain of glucoses. And if we think about it like the previous image of the glycogen with that highly branched structure, the same situation applies with the enzyme. It can only break down one glucose at a time. So it can take this one, and then it can move on to the next one, and then the next one, and then the next one, and the next one. It doesn't have multiple branches in order to break down lots at once. So starch is a time release energy. You've probably heard of carbo loading, which is when people eat a lot of pasta before they run a marathon or go on a long bike ride. And that's because they're building up those starch stores so that they can be broken down over time over their long run or ride or whatever strenuous thing they're doing. So they can actually be up to 10,000 molecules of these. And these are what we call also polysaccharides. So complex carbohydrates are also called polysaccharides. Let's go over here. And poly means many. So many sugars at once. So if we take a look at this image from your text, figure 3.5, I really like this image because it demonstrates exactly what they mean by time release. So this blue curve here, as you see, it has a peak that's pretty quick. If time moves to the right, then we see this peak happen early in foods high in simple sugars like glucose and fructose and sucrose even. And foods high in complex carbohydrates, so starchy things. This is hummus made out of chickpeas or garbanzo beans. It is a time release. We see not a very steep curve, but over time it continues to release glucose molecules rather than the crash that comes with simple sugars and sweets. Let's talk now about structural polysaccharides. So not all polysaccharides are used for fuel. Sometimes they're used for building blocks. For example, up on the top here we have something called cellulose. Cellulose is made of glucose molecules, so interestingly, but we see that there are hydrogen bonds between the parallel chains. So if we were looking for hydrogen bonds, they would be between the chains, holding them together and making this rigid structure. So it's rigid, but it's also flexible. So it's what wood is made out of, and cotton, and it's what we call fiber in foods. It's indigestible. we can't digest it. The second type of structural carbohydrate is what we call chitin, and it's spelled C-H-I-T-I-N. It's pronounced like K-Y-T-I-N, chitin, not chitin. So these are also linked glucose molecules, but instead they have hydrogen bonding not just between them, but also within these chains. So it's a more complex structure, and it's extremely hard and strong. And this makes up insect exoskeletons, and also the cell walls of fungi. So let's quickly go over the carbohydrates. So we have simple and complex. The simple, they include single subunits, glucose is our most important one. And complex. Now complex has a couple of categories. So we have digestible and indigestible. And digestible includes starch and glycogen and disaccharides. So disaccharides and polysaccharides. Disaccharides include things like sucrose, lactose, and polysaccharides which are things like glycogen and starch. And our indigestible ones are cellulose and chitin. And in all of these carbohydrates, we get the energy from breaking carbon-hydrogen bonds. So these are, they have C, H, and O, break bonds to get energy. The second type of macromolecule I want to discuss are lipids. Now, they don't have any particular subunits like the carbohydrates did, being, say, glucose, repeating units of glucose. They don't really have anything like that, but they do come in many different forms and all have a few of the same characteristics. For example, they are non-polar. We talked in chemistry about how water is a polar molecule, meaning it has a slight positive charge in some areas and a slight negative charge in others. So the oxygen having slight negative, hydrogen having slight positive. Well, lipids don't have anything like that. They have an even distribution of electrons, and so they don't have any sort of charge on them by themselves. So they don't dissolve in water at all. They are greasy to the touch, so they'll be oily and slick. And they also can be a significant source of energy storage. So we're going to talk about these three major types. So we have the fats, which are long-term energy storage and insulation in animals, and some in plants, but not as much. And the sterols and the phospholipids. Those are the three types we'll talk about. So dietary fats are what we call triglycerides. If you go to the doctor and you have your blood drawn, you have your triglycerides check. They are checking for fat in the blood. Now it has two major parts. The top portion in this image is what we call a glycerol molecule. 3 carbons connected to oxygens, and connected to those oxygens are what we call fatty acid tails. Each of these is a long carbon chain, and it kind of looks like a tail, and we call it a fatty acid because it has acidic properties. We'll talk about acids and bases later on, but it's a fatty acid. And there are 3 of them connected to the glycerol molecule. So we call it a triglyceride, meaning 3 things attached to a glycerol molecule. So fat contains more energy than sugar. So sugar has about 4 calories per gram, but fat has about 9 calories per gram. So it's over twice as energetically dense. It means we can get more energy from 1 gram of fat than we can from sugar. Now, dietary fats include what we call fats and oils. Now, interestingly, humans have actually evolved to enjoy the taste and the feel of fat because it's energetically favorable. We get more energy from fat than we do from sugar. And that has been evolved in us because for a really, really, really long time, millions of years, humans, or human-like pre-humans, they were mostly gatherers and hunters and foragers, and so they ate what they could. And the ones who were eating more fat were more likely to survive. And so a taste for that was sort of selected for. We'll talk lots about natural selection, and you might have a better understanding of what I mean there later on. But survival is really the key there. There's more energy in fat, so we ate fat because there was more energy. There are 3 types of dietary fats I wanted to discuss with you. The first is called a saturated fat. Now, the reason why we say it's saturated is because it has, attached to these carbon molecules, it has the maximum number of hydrogens on each one. And so remember that carbon can make 4 bonds. And if it has a carbon on either side, then it can hold up to 2 hydrogens. We'll see why this isn't true for unsaturated in a minute. But if we have these carbon chains, and all of those carbons have hydrogens, 2 hydrogens, then we call it saturated. A saturated solution is something that has as much as it can hold. So in saturated fats, interestingly, they can stack on each other like this. They can pack together really, really compactly because these fatty acid chains will just lay right on top of each other. And as a result of that, saturated fats are solid at room temperature. So if you want to know if something is saturated or unsaturated, just look at it at room temperature. Now, these can also be called animal fats. We find these mostly in animals, and they are not essential for our health. We don't need saturated fat at all. The other type is unsaturated fat. So an unsaturated fat has a slightly different structure. Now notice here that these tails, instead of being nice and straight, let's draw like the book had here. Instead of being nice and straight, they have kinks in them. Those kinks are caused by double bonds, and it's just the geometry. If you ever take chemistry, you'll understand more about that, but we're not going to concern ourselves with it today. So if we have a carbon chain with a double bond in it, it changes the shape. So a double bond is actually going to be like this. It's going to change the shape to be bent. So remember what I said about carbons having as many hydrogens as they can hold, and that maximum number in a carbon chain is 2? Well, there are 2 carbons here that can only hold 1, because carbon can have a maximum of 4 bonds. So this carbon here has 1, 2, 3, 4. And because it has 2 to this carbon here, it can only have 1 hydrogen. This is unsaturated. This particular carbon is unsaturated. And so is this one there. They cannot hold 2 hydrogens because of the double bond, and that's why we call them unsaturated. Now, unsaturated fats, because they have these kinked type of hydrocarbon chains, the fatty acid tails, they actually are liquid at room temperature, not solid. So they are what we call oils. So over here on your image it says they are liquid at room temperature. So olive oil, canola oil, fish oil, any other type is going to be unsaturated, an unsaturated fat. Now these we do actually need for our health in some capacity, not a ton, but some. So we can also find these things in things like avocados and peanuts and fish. Oh, here comes a cat. There she is. No, sorry, you can't hang out with me right now. Sorry. A third type of fat is called trans fat. I'm sure you've all heard of this, but this is an artificially saturated fat. This is produced by a process called hydrogenation. If you read somewhere that says partially hydrogenated, it's a trans fat. It says like partially hydrogenated canola oil or palm seed oil or anything. Those are trans fats, and they have a very different structure. Take a look at the image on the left. So remember how we have our carbon-carbon double bond, and it makes these kinks like this that have an angle here. Well, trans fat does something different. The carbon bond actually goes like this, and the carbon goes up instead of down, and it has more of a chair shape with 2 angles in it rather than just 1. So the orientation, the reason they're called trans fats is because in organic chemistry this is the orientation of it. It's a trans double bond. Don't worry about that. So it's unusual as far as the body does, as far as the body knows, and it doesn't break it down as often. And like saturated fat, it can accumulate in the blood vessels and cause obstructions. And there's actually been studies of trans fats and how consuming them actually really does increase the risk of heart disease. They did a study on nurses and found that just a 2% increase in trans fat consumption resulted in a 2 times more risk, twice the risk for heart disease. So adding 2% you get twice the risk of heart disease. So it is actually a bad thing that you probably should be aware of and consume sparingly. The second type of lipid I want to mention are sterols. So sterols are lipids, but they are not fats. They are specifically sterols, and that's because of their shape. And these are also not for energy storage. They have different functions. Instead, they regulate certain growth functions and sexual development. So included in those are cholesterol, estrogen, and testosterone. So these are examples of sterols, and here are their specific structure. They are 4 rings bound together in this structure, and they have different what we call side chains, things that make them slightly different from one another. So notice, interestingly, how similar estrogen and testosterone are. Extremely similar, but they have very, very different functions in the human body, at least. Cholesterol, though, is actually essential. We need a little bit. However, most of it is made in the liver. About 90%, so we really do not need to consume very much cholesterol, because we make most of what we need, especially for cell membranes, and we'll talk about that more in the next couple chapters. The third type of lipid I want to mention are the phospholipids, and I'm just going to cover what they're made of here and really their basic function, because you're going to know more about them later on when we talk about the cell membranes. So they are the major component of cell membranes, and as you see, the symbol is very similar to that of the triglyceride, if you recall, like this, except there are only 2 fatty acid tails on the phospholipids. So it does have a head area up here at the top, and it has, instead of a glycerol, it has what we call a phosphate group, which is a phosphorus and 4 oxygens. Those of you who had chemistry know about the phosphate group. And interestingly, it groups itself like this to make cell membranes. And we're going to talk about that lots until you're probably sick of it. So the final type is waxes. Now, waxes, you probably already know this, but waxes repel water. And they are incredibly hydrophobic. They do not touch water at all. They don't want to be near it. Water will literally beat up on them and just roll off. That's what happens when you wax your car, or when you even apply Rain-X. It's kind of like that to your windshield. And interestingly, it's a glycerol molecule. So we have this carbon, carbon, carbon with oxygen, 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 but with only one really long fatty acid tail. And waxes are not only for cars. They are also found in animals and plants. So in plants, leaves have a waxy coating on the top to prevent water loss. And same with insects, they have sometimes a waxy coating on their shells, and also feathers. Bird feathers have certain types of waxes to keep them from getting waterlogged if they get wet. So if we recap lipids, we had 4 types. We have the triglycerides, we had the sterols, phospholipids, and waxes. So triglycerides are our fats. We have 3 types: saturated, unsaturated, and trans. And saturated is really the only one that we need. Sterols are for their hormones. Let's do that in the orange color. And for regulation of various processes, and for the membrane. Phospholipids are the main component of membranes. And waxes are coverings that prevent water loss. And water penetration, I guess? Water contact. So there are our 4 types of lipids: triglycerides, sterols, phospholipids, and waxes. Let's move on to our 3rd type of macromolecule. Our 3rd type of macromolecule is protein. Now, protein has a huge amount of diversity, and they do all kinds of things within the cell and the body in general. And they are the major building blocks, and they do most of the work of living things. They can be structural. So things like hair and fingernails and feathers, horns, cartilage, tendons, these are all made of protein. Our hair is made of protein. Our fingernails are made of protein. For protection, so they actually aid in fighting off microorganisms and keep you from bleeding out if you ever cut yourself with your razor. Regulation, they even control cell activity at times and constitute some hormones. So not all hormones are sterols. For contractions, for muscle movement, heart pumping, and then even for sperm to swim. And transportation, they are carrier molecules. I realize that my little picture, my video here, is blocking this, but they carry molecules throughout the body. This, for example, is hemoglobin, the protein that carries oxygen throughout the body from the lungs through the blood. Proteins are made up of subunits called amino acids that are also sometimes called peptides. And it consists of, amino acid in general consists of 3 parts: an amino group, which has a nitrogen in it, a carboxyl group, which has 1 to 2 oxygens depending on what it's bound to, and a side chain. Now there are 20 different side chains, and they all have different compositions and structures. For example, down at the bottom we have, for example, glycine. Glycine is our simplest side chain with just a single hydrogen. Alanine has what we call a methyl group attached, which is a CH3. And tryptophan, down at the bottom, this is the most complex of them, which has a double ring structure, which even includes a nitrogen. So when they're bound together, you will see it'll be nitrogen, carbon, carbon, if we're going from top to bottom here. And then they will bind again, and it will repeat: nitrogen, carbon, carbon. So on this nitrogen, we will have hydrogens. Now nitrogen only likes 3 bonds, so it'll only have 1 on this one. And then we'll have our carboxyl groups, which will have a double bond oxygen. And this will have a double bond oxygen and OH. And then over here, we will have our side chains, which are whatever they are. So here we have a special bond between these, and we call this a peptide bond. And more than one peptide is called a polypeptide. That's another word for protein. Now these amino acids are kind of like an alphabet. So they can occur in any sort of arrangement, kind of like our language. Some letters we see together more than once, some we don't see very often at all. And the sequence means we get a different protein for each different sequence. So in protein in the diet, we have 2 different types. So we have complete and incomplete proteins. Now complete proteins include all of what we call essential amino acids. So essential amino acids we cannot make ourselves. We have to get them from the things that we eat. And not all foods have everything. So if you just say, I need 50 grams of protein a day, well, you probably need 50 grams of what we call complete protein, meaning it has all of the amino acids, either by one type of protein or a combination of them that will give you the complete amino acids. For example, complete proteins include ones from milk, tofu, soy milk, which is tofu and soy milk, that's both soy, chicken, and beef. So all of these are complete. Notice that these are filled in, which means they have all of these essential amino acids that we can't make. We have to eat them. Now, notice that apples and almonds, for example, they are missing a few of the essentials here. But sometimes you can eat combinations of things and still get what you need. For example, rice with lentils. That's a very popular dish in India, having rice with lentils. And just because we have a deficiency here, we can still get it from the rice in this methionine and cysteine. If we have a lysine deficiency, we can get it from the lentils. So you can have complete protein content even if you're eating more than one different type of protein. But it's really quite important, especially if you're vegetarian. You need to be aware of what kind of protein you're eating, because you could be eating a deficiency in one of the types of essential amino acids. Proteins are particularly special because their function is dictated by their 3D shape. Their shape is essential to their function, because if they lose that shape, then they can't work anymore. But there are 4 levels of organization of proteins. And I'm just going to cover these really briefly, but you should know a little bit about what they mean. So in this primary structure, it's simply the sequence of amino acids. So if we have alanine, lysine, cysteine, methionine, cysteine, just that sequence, each one, these together, that's the primary structure, like beads on a string. The amino acids connected by peptide bonds. Now, these strings can also arrange themselves in different ways. Because they have different side chains, they can actually hydrogen bond, these kind of weak temporary bonds, to different pieces of each other. And they form things called alpha helices and beta sheets. You don't have to know that. But they look like sometimes these flat areas, kind of like this, or sometimes they're little curlicues. So it's twisting and folding on itself. It's still only one protein, but it's twisting and folding on itself. The tertiary structure means it's a complex 3D shape because of all those interactions of the chains and of the environment. So it's a weird globular kind of structure. And quaternary structure means that 2 or more polypeptides will work together, and that happens a lot in biology. These polypeptides will bond together and work together. Now, like I said, if a protein loses its shape, then it doesn't work anymore. And that is a process called denaturation or denaturation. And it happens from extreme environments. So for example, temperature, like if you heat something up, or pH disruption. So heat from, say, like cooking, and pH from the stomach can denature these proteins. And that means that they basically unravel and kind of fall apart. So think about cooking an egg. When you cook eggs, if you've ever cooked or seen an egg cooked, they will turn white instead of clear. There's a ton of protein in eggs, and the heat from cooking denatures that protein, and it all kind of unravels and sticks together. So those proteins in the eggs that you cook can no longer function like they did in the egg. Now they are food for us, and we can use them and break them down to get their amino acids. Now, something interesting I like that your book brought up is curly and straight hair. Now, hair texture is genetic. You get that from your parents, and there's nothing you can do about that. And it's because of tertiary interactions. Of the proteins in your hair, because your hair is made of protein. So the 3D structure of your hair proteins determines if you have nice straight hair or if you have lovely curly hair, or even something in between. I have wavy hair myself. So it's not really curly, it's not really straight, kind of in between. So the way you can change your hair style, and the reason why it kind of resets when it's wet, is because of hydrogen bonding. Because hydrogen bonding is between hydrogen of one and nitrogen or oxygen of another molecule. So water makes tons of hydrogen bonds with your hair. And when it dries and evaporates, those hydrogen bonds are broken, and instead of having water and a protein, now you have pieces of your proteins hydrogen bonded together. So it kind of resets the shape. So kind of interesting. So enzymes are particularly important in doing cell functions, so making things work. So what they do, they initiate and accelerate chemical reactions. So initiating means they get them started, because usually chemical reactions need some sort of push, a tiny bit of energy to get started. Think about somebody sitting at the top of a slide. You give them a little push and they can go down the slide. Now, some reactions make energy, some produce energy and some consume energy, but they still need some sort of little push to get over that hill. So what enzymes do is that they reduce that energy needed. You don't have to give them as hard of a push to get them going. But enzymes are kind of cool because they're extremely specific. And so this enzyme here, this is the lactase enzyme. If anything ends in ase, it's an enzyme. So amylase, or protease, or DNase, or RNase, anything that ends with ase is an enzyme. So this one is lactase, and this is lactose, the sugar, and it's what we call a substrate. It's the thing that fits right in here in the active site. It's a remember that the proteins are weird 3D structures, and they have a special pocket where their substrate fits. Exactly. And very few other things will fit. Usually nothing else will fit. That's why they're so specific. Only that one thing can fit. So lactose will wiggle itself right into that active site, and kind of like a lock and a key, it starts the reaction, and lactase breaks apart that disaccharide, which is right here. Breaks it apart here to form galactose and glucose, which is what lactose is made of. So this is just one example, but there are lots of different examples of enzymes. And something that's cool about enzymes is that they reset themselves. They can be used over and over and over again, hundreds and thousands of times. They aren't just used up when you're finished. So if we do a quick recap of proteins, they are made of amino acids. They have 4 levels of structure. They are building blocks and heavy lifters of the cell, and they include enzymes, which speed up or start reactions. So let's move forward. The final type of macromolecule I want to talk about are the nucleic acids. So nucleic acids are made of repeating units of what we call nucleotides. And nucleotides have 3 distinct parts. They have a sugar, they have a phosphate, and they have a base. And they repeat. Like this. So we have a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, etc., to make what we call the backbone. And we have 4 different bases. Let's do that one in a different color. The 4 different bases generally are adenine, thymine, guanine, and cytosine. But the basic structure is the sugar phosphate backbone. And in the middle we have the bases. So on the left, if we look at the figure, there's the backbone, and sticking out are the bases made of nucleotides. The first type of nucleic acid I want to talk about is deoxyribonucleic acid. This is DNA. And this is the thing that stores our genetic information. And its structure is that of what we call a double helix. It's like a twisted ladder, like they took a ladder structure, like a rope ladder maybe, and twisted it around so that these 2 parts are twisting around each other. So twisting and twisting and twisting. And the structure is the backbones are on the outside. There's one and the other. And they're joined together by the bases in the middle, which are connected by hydrogen bonds. Here we are again with more hydrogen bonds. And so we call these base pairs. And we call them base pairs because they pair with each other. Adenine, which we abbreviate to A, will only ever hydrogen bond with thymine. Thymine will only ever hydrogen bond with adenine. Cytosine, which we abbreviate to C, will only ever hydrogen bond to guanine, G, and vice versa. Guanine cannot hydrogen bond with adenine or thymine. Cytosine cannot go with adenine or thymine. Adenine can't go with guanine, cytosine, etc., etc. So we call these complementary base pairs. Sorry, we're back. So they're not complementary base pairs, like they don't think that they look nice today. It's complement, and a complement just means it is a pair. It's something that goes with it, goes together. So those things only ever bond with each other. A to C, A to T, and C to G. So interestingly, almost every human cell, every liver cell, every heart cell, every lung cell, every brain cell has all of our genetic information in it. There are very few exceptions. For example, red blood cells don't have, our genetic information, but all the others do. And we humans have more than 3 billion base pairs. So our DNA is very, very, very long. It's a ton of information that is stored. When we talk about biotechnology, we'll talk about the Human Genome Project and what a huge undertaking it was to figure out all of the genes that humans have. So again, we'll talk more about DNA in specific in a few chapters. The next type is ribonucleic acid, or RNA. Now there is a difference there are several differences between DNA and RNA. First of all, the sugar is different. So instead of deoxyribose, this one is called ribose because it has one extra oxygen, and deoxy for DNA means that oxygen has gone away and been replaced by a hydrogen. But in this case, there is an additional oxygen. So ribose is the sugar. Now, also in RNA, thymine that normally bonds with adenine is replaced by something called uracil, which is very similar in structure and still will create a double bond with adenine. So it's C with G as usual, but instead it's A with U, and I don't even have a pink down here. We'll do red instead. And then U with A, G with C, which is orange, and A with U again. Of course, my picture is covering it up. So uracil is different, and RNA is also single-stranded. It's not a double helix. It is a single strand of nucleic acid here. So it can be folded up on itself, and it actually can have some really cool, funky little shapes like this. We studied some when I was in my molecular biology classes. But yeah, it can look kind of like that. But it doesn't have a double helix like DNA does. Instead of RNA being information storage like DNA, it's more like it transfers information. It's the middleman between DNA and protein. We will talk much more about that when we talk about protein synthesis. But it helps direct the building of the protein. It's what part of the cell reads in order to make that protein. Again, we will have many more details later on. So as our last little hurrah here, let's quickly compare DNA to RNA. So DNA, we know it stores genetic information. Its sugar is deoxyribose. It has the bases A, T, C, and G. That's adenine, thymine, cytosine, and guanine. And it's double-stranded. RNA, on the other hand, it transfers genetic information. It has ribose as a sugar. The bases are A, U, C, G. That's adenine, thymine, uracil instead. And it is single-stranded. So, quickly about nucleic acids. Just to recap, they are made of nucleotides. Remember that nucleotides have a sugar, a phosphate, and a base. The two types of nucleic acids are DNA and RNA, and they have different functions as we just discussed. So that is the end of this video. I realize it's an hour long, but I couldn't really shorten it anymore. Thank you so much for watching. Please do the quiz after this. There will be a link right under this video on Blackboard. Have a great day. Thanks for watching.
Not good at walking in straight lines for like the first 20